Cell physiology and pathophysiology. Disorders of membrane transport, cell signaling, and cell death include the conditions cystic fibrosis, which has an altered ion channel, familial hypercholesteremia, which has a receptor-mediated endocytosis, receptor-targeting autoimmune disorders such as myasthenia gravis and Graves' disease, and cancer, which is a failure of apoptosis. As a clinical nurse specialist, you should know that common medications to reduce heartburn block histamine receptors or active transport of hydrogen ions with protein pumps. Some cardiovascular disorders are treated with calcium channel blockers, and selective serotonin uptake inhibitors that are used to manage depression and anxiety can block a secondary active transport protein. Pathophysiological alterations at the cellular level underlie many diseases and medications to treat those disorders that often target specific cell proteins and functions. The unique functions of the organs and systems of the body are based on the differentiated structure and functions of the cells making up those organs and systems. The cytoplasm refers to the entire contents of a cell. The aqueous intracellular fluid contains dissolved electrolytes, small biomolecules and metabolites, high-energy phosphate compounds, and key intracellular proteins. Membrane-bounded organelles include the nucleus and mitochondria, each with two outer membranes, the rough and smooth endoplasmic reticulum, the Golgi apparatus and vesicle budding from the Golgi, lysosomes, peroximones, and vacuoles, ribosomes, centrosomes, and protein filaments do not have membranes. The boundary between the intracellular and extracellular fluids is the cell membrane, and also it is known as the plasma membrane. The cell membrane is a phospholipid layer where the outer leaflet phospholipids are oriented with their polar head groups facing outward toward the extracellular fluid. The inner leaflet phospholipids are oriented with their polar head groups facing the intracellular fluid. Molecules of cholesterol are interdispersed with the phospholipids controlling membrane fluidity. Integral proteins cross the membrane, and some of these function as receptors, others as transporters or channels. Peripheral proteins are often attached to inner leaflets and work with integral proteins to alter the cell function. The outer leaflet is rich in glycoproteins and glycolipids with complex branching carbohydrates attached. The layer of sugars projecting outward from the cell is referred to as the glycocalyx, and its function in the cell is cell recognition. The membrane is dynamic, moving with modal cells such as white blood cells, conducting exocytosis and endocytosis, and enlarging with cell hypertrophy. The cell nucleus holds most of the cell's DNA, and the remainder is found in the mitochondria. Unless a cell is preparing to divide by mitosis, the nucleus contains DNA organized in chromosomes that are wound around histone proteins. The nuclear membrane has two layers with relatively low permeability. Red blood cells do not contain nuclei, and skeletal muscle cells contain multiple nuclei. Only these two exceptions, therefore the rest of the body's cells contain one nucleus. A ribosome is made of large and small subunits that attach to messenger RNA for the purpose of protein synthesis. The rough endoplasmic reticulum is the site of chaperone proteins that detect the progress of protein folding and that mark misfolded proteins for destruction. The smooth endoplasmic reticulum is the location of some steps in steroid hormone synthesis in certain endocrine gland cells. It is also the site of synthesis of new membranes that can refresh the cell membrane or organelle membranes. This reticulum is a major site of cellular calcium homeostasis, where sequestering calcium to keep cytoplasmic free calcium levels low most of the time and release calcium in response to the membrane receptor. The Golgi apparatus functions to sort, modify, and package proteins and lipids that are delivered in vesicles from the near endoplasmic reticulum. It also forms other organelles that are vesicle-type organelles, including lysosomes and secretory vesicles. The mitochondria are the energy of the cell and produce much of the cell's adenosine triphosphate, or ATP. Remember that ATP is the energy source for chemical reactions and for ion transport in the cell. Lysosomes contain degradative enzymes that break down aging cell organelles and molecules. These enzymes are in the class called hydrolases, which depend on low pH for their function. Peroxisomes are enriched in enzymes that use oxygen for their reactions. The cytoskeleton is made up of protein filaments that extend through the cytoplasm and giving the cell its shape. Organelles and vesicles can move along these filaments, and in mobile cells like white blood cells, they are responsible for cell movement. Oxygen, carbon dioxide, and steroid hormones, as well as other lipid-soluble substances, are able to freely diffuse across the membrane. Ions and biomolecules like glucose and amino acids require specific membranes and mechanisms to cross the membrane. Sodium is high extracellularly and potassium is high intracellularly. Simple diffusion describes movement across a barrier without a specific transport protein. Diffusion occurs down a concentration gradient from an area of high concentration to an area of low concentration. Here, this method of transmembrane movement is used by small nonpolar molecules like oxygen and carbon dioxide, and by lipids such as fatty acid and steroid hormones. The concentration gradient across a membrane permitting movement from high to low concentration or downhill movement is exhibited in B. The concentration gradient across a barrier or a solute that needs energy to be moved from a region of low to high concentration that requires the ATP is exhibited in C. In endocytosis, a portion of extracellular fluid is engulfed by the cell membrane into a vesicle that pinches off and then enters the cell. The endocytosis of small amounts of extracellular fluid is known as pinocytosis. The process of exocytosis is basically the reverse of endocytosis. A membrane-coated vesicle moves to the cell membrane and fuses with it. Then it releases the contents into the extracellular fluid. Regulated exocytosis is a process where a molecule or protein that is destined for secretion is held in the intracellular vesicles until a specific event triggers exocytotic release. Endocytosis and exocytosis are mechanisms for bulk movement of a large number of solutes or large solutes like proteins across the membrane of the cell by means of membrane-bound vesicles. Movement of polar and hydrophilic substances down their concentration gradient is facilitated by a membrane protein transporters. These transporters bind a molecule of the substance on the side of higher concentration and then release it on the side of lower concentration. Because this movement needs a protein carrier, it is known as to be facilitated. Active transport is a mechanism for moving a solute against the gradient concentration that requires the input of energy, or ATP. There are two types of active transport. There's primary and secondary. The most abundant primary active transporter is the sodium-potassium pump in the body. Another name for the protein of calcium ATPase is sarcoendoplasmic reticulum calcium ATPase. This pump is found on cell membranes and on the membrane of the endoplasmic reticulum. Based on the sodium gradient that is determined by the sodium-potassium pump, cells have various transporters that link downhill sodium movement into the cell to drive the uphill movement of other substances against the concentration gradient. This process is known as secondary active transport because it does not use ATP. Aquaporons are channels that serve as major transport mechanism for water to move in and out of cells under the influence of osmotic pressure. This is regulated in kidney cells in the distal nephron, where water absorption is controlled by the hormone vasopressin. When the ion channels are open, the ions will freely diffuse through until the pores close or until the ions reach electrochemical equilibrium based on the concentration difference of the ion and the membrane voltage that develops due to the consequent movement of the ion. The presence of ion channels in the cell membrane contributes to the ability of cells to develop a resting membrane potential, a voltage difference across the cell membrane. Most cells of the body have a resting membrane potential that is negative. What causes this membrane negativity is the potassium leak channels outnumber other channels at rest, so that the potassium movements are the greatest factor setting the electrical equilibrium. The composition of the extracellular fluid and intracellular fluid differs regarding the distribution of sodium, potassium, chloride, and calcium, so that each of these has different gradients for movement when the ion channel opens. There is constant activity of the sodium-potassium pump. This maintains the proper ion composition of extracellular and intracellular fluids. The action potentials of neurons are the basis of the brain activity, and the action potential of muscle cells are the basis of contraction of the skeletal and cardiac muscles. Mutations in ion channel genes are the source of many disorders of neuron and muscular function. An example is epilepsy. Many rapid homeostatic adjustments are made possible by the autonomic transmitters norepinephrine in the sympathetic nervous system and acetylcholine in the parasympathetic nervous system. Hormones like epinephrine, vasopressin, glucagon, and pituitary trophic hormones have rapid action to modulate activity of their target cells, glands, and organs. The action of all these ligands are mediated by binding to membrane G protein coupled receptors. Many of these reactions are reversible once the ligand diffuses away from the receptor, and there are several other mechanisms that can stop or reverse the actions of receptor activation, therefore allowing compensatory adjustments to return to normal after a time of altered activity. The autonomic nervous system is the major regulatory system of the body. Autonomic neurons located within the central nervous system consist of preganglionic neurons that use the transmitter acetylcholine. Parasympathetic postganglionic neurons are cholinergic, releasing the neurotransmitter acetylcholine, and most sympathetic postganglionic neurons are adrenergic, releasing the neurotransmitter norepinephrine. Found on all of the cells, these G protein coupled receptors are the most common mechanism by which hormones and neurotransmitters influence cell and organ function. Ligand binding to a stimulatory AC receptor causes the dissociation of the attached G protein, allowing the AX subunit to move to and activate the enzyme acetylcholine. The beta-Y subunits remain bound together but can move within the membrane to modulate the activity of other membrane proteins. Stimulatory and inhibitory AC-linked receptors. The AC can be stimulated or inhibited depending on the type of receptor activated. Stimulatory receptors like the R-stim work through the GS to increase the AC activity and cyclic adenosine monophosphate production. Inhibitory receptors work through the GI to decrease the AC activity and the C-amp production. A protein kinase is an enzyme that adds a phosphate group onto target proteins. This process of phosphorylation occurs on the hydroxyl containing amino acids serine, threonine, and tyrosine. The beta-adrenergic receptor activation is the mechanism by which the sympathetic nervous system increases the heart rate and the force of contraction. The presence of both stimulatory and inhibitory types of receptors for the adenyl cyclase system provides for homeostatic adjustments that can be up-regulated or down-regulated for adaptive changes. Activated phospholipase C splits the membrane, with DAG remaining in the membrane and IP3 diffusing into the cytoplasm, thus serving as an intracellular secondary messenger. The IP3 binds to receptors on the endoplasmic reticulum, causing the endoplasmic reticulum to release stored calcium in a burst. This increases the cytoplasmic calcium concentrations. When the sympathetic nerves innervating the blood vessels release norepinephrine, it binds to these receptors, and this increases the intracellular levels of IP3 and releases the calcium from the endoplasmic reticulum. Calcium elevations lead to calcium and calmodulin binding and the activation of an enzyme that causes cell contraction and ultimately producing vasoconstriction and increasing blood pressure. Hydrolysis of the GTP by the G proteins limits the duration of the hormone receptor actions. This is similar to a timing switch. In A, there are no ligand that binds the receptor. The receptor is inactive and the G protein is bound to GDP. In B, the hormone binds to the receptor. The GDP is now replaced by the GTP, and the G protein dissociates into the alpha and the beta Y subunits. In C, the alpha subunits activate the target enzymes and proteins. In D, the alpha subunit is intrinsic GTPase activity splits the GTP. Therefore, it binds the GDP and the beta Y subunits and returning to the inactive state. The cyclic guanosine monophosphate, or cGMP, is synthesized in retinal photoreceptor cells and plays a critical role in vision. The GMP also aids in vascular smooth muscle as a potent vasodilator. Many of the receptors that are used for growth, development, and rapid cell proliferation have intrinsic enzyme activity rather than requiring the G protein to activate an enzyme. Most of these enzyme-containing or enzyme-linked receptors have tyrosine kinase activity, meaning that the phosphorylate target proteins at the tyrosine sites. Most of these receptors have their greatest activity during periods of rapid growth and cell proliferation, embryonic and fetal stages, and during childhood. The dysregulation of tyrosine kinase signaling pathways is common in cancer, fibrosis, chronic inflammation, and disorders of immune hyperresponses. Intracellular signaling is initiated by receptors using tyrosine kinase mechanisms. Number one, these receptors are present in monomeric form, meaning that they are single, non-interacting chains, and are inactive with no extracellular signal. In two, binding of ligands causes the formation of dimer with two receptor molecules coming to close physical proximity and the tyrosine kinase regions of each phosphorylating the other chain. In three, once phosphorylated, the intracellular portion of each receptor adds more phosphates outside the tyrosine kinase region. In D, the newly phosphorylated sites become scaffolds where other signaling proteins numbered 1 through 3 in the figure can be phosphorylated, and exert their own enzymatic activities. Some of the tyrosine kinase type receptors will stimulate cell proliferation and are implicated in wound repair and in cancerous transformation of cells. Epidermal growth factor receptor activation turns on several signaling pathways that ultimately alter gene expression, transcription, and translation to increase rates of cell division. When it is initiated in an unregulated fashion in adulthood, this can promote cancer. Cytokines are protein messengers of the immune system. Immune responses to foreign proteins include rapid proliferation of clones of immune cells, which are monocytes and lymphocytes, to help fight off foreign invaders. Although classically associated with cytokine signaling and immune responses, the Janus kinase and STAT proteins are also mutated in some cancers and are the target of some anti-neoplastic medications. Transforming growth factor beta is implicated in pathophysiological induction of fibrosis in a variety of diseases, including the liver, lung, blood vessels, and various tissues. Steroid and thyroid hormones are relatively nonpolar and hydrophilic. These are able to move across the cell membrane by simple diffusion. Once inside the cell, there are one of two pathways that are followed. Some of the steroids will bind to the receptors in the cytoplasm and are then transported into the nucleus, where other steroid hormones and thyroid hormones will diffuse across nuclear membranes to bind to receptors already inside the nucleus. No matter what, the hormone receptor complex ultimately binds to regulatory elements on DNA and alter transcription and translation within that cell. Short-term adaptations mediated by autonomic and endocrine signals often act by way of the GPCRs with rapid onset and several mechanisms of signal transmission. Intermediate and long-term adaptations can be mediated by steroid and thyroid hormone receptors that alter gene expression and protein translation. Some normal and pathophysiological adaptations alter the activity of the receptors that are involved in growth and development, promoting cell proliferation that can be beneficial, like the immune response, or pathophysiological, which would be immune hyperactivity or cancer. There are three types of muscle cells in the body: skeletal muscle, cardiac muscle, and smooth muscle. The sarcolemma refers to the cell membrane, sarcoplasm to the cytoplasm, and sarcoplasmic reticulum to the endoplasmic reticulum. The microscopic appearance of skeletal and cardiac muscles show a striated appearance, dark bands alternating with light bands. The repeating unit is the sarcomere, the functional unit of muscle contraction. Skeletal and cardiac muscle cells shorten by forming cross bridges between actin and myosin filaments that are highly organized into sarcomeres that shorten the unit. During muscle contraction, myosin heads bind to recognition sites on actin bands and pull the actin bands toward the center of the sarcomere, using sarcomere shortening and ultimately shortening the whole muscle. Genetic mutations of some of these connecting proteins are responsible for the hereditary muscular dystrophy syndromes. Troponin C is the site of calcium binding under conditions of muscle contraction. Sarcomere shortening due to myosin-actin cross-bridge formation. The action of many strands of myosin and actin form cross bridges that adds to the shortening of all the sarcomeres in a muscle fiber, producing contractile force and movement. The triad of structures regulating muscle calcium release. Invaginations of the plasma protein, T-tubules align next to terminal cisterns of sarcoplasmic reticulum. When the muscle action potential is propagated along the membrane and down the T-tubules, the signal is conveyed to the sarcoplasmic reticulum to release its calcium stores. The DPH receptor of the T-tubules are adjacent to the RYR of the SR cistern depicted in A. In B, the membrane depolarization due to the muscle action potential causes the DHP to activate the RYR, and the SR releases its stored calcium. The wave of calcium quickly diffuses around the actin filaments binding to troponin, shifting troponomyosin and allowing myosin-actin cross bridges to form. Sarcomere structure and the striated appearance are quite similar between the skeletal and cardiac muscles. Skeletal muscles carry out involuntary reflex and voluntary activity that is directed by the nervous system, and their contractions are initiated by motor neurons in the spinal cord and the brainstem. Skeletal muscle fibers are elongated and organized into progressively larger bundles that ultimately transition to tendons that join bone to muscles, allowing purposeful movement. Skeletal muscles are activated in sequence to create a series of movements. The heart contracts autonomously in response to the pacemaker cells of the sinoatrial node. Cardiac cells are short and linked mechanically and electronically to neighboring cells that surround the cardiac chambers. Contraction compresses and squeezes the chambers, ultimately leading to ejection of blood into the vasculature of the body or the lungs. The cells in the atria and the ventricles of the heart contract almost simultaneously for efficient ejection. Smooth muscle cells are found in the walls of hollow organs like the GI tract, the bladder, and the uterus, and they make up contractile layers of blood vessels and airway. Contraction and relaxation of vascular and organ smooth muscle layers are primarily regulated by the autonomic nervous system and by local and circulating chemical mediators. Contraction of smooth muscle in A, it is relaxed, and B, it is contracted. In the smooth muscle cells, actin-myosin strands form a loose meshwork crossing the cell. The strands are anchored by attachments to the plasma membrane and to dense bodies within the cytoplasm. When the calcium increases, the cell shortens. Muscle cells are contractile cells that shorten upon stimulation, providing force for body movements, heart pumping, and contractions of the walls of blood vessels, airways, and internal organs. Actin-myosin cross-linking and cross-bridge cycling are responsible for muscle contractions, with cross-bridge formation and cycling also requiring calcium ions and ATP. General mechanisms for activation of vascular smooth muscle. Electromechanical coupling is shown on the left. A voltage-operated calcium or Ca2+ channel allows calcium entry upon cell depolarization. Pharmacomechanical coupling is shown on the right. A receptor-operated Ca2+ channel allows calcium entry upon activation of various receptors. Other receptors will increase the calcium through the phospholipase C system and IP3. Many cells have finite and short or intermediate lifespans. Examples are white and red blood cells, skin cells, and cells that line the GI tract. As these die, they must be replaced by stem cells that are self-renewing and then give rise to daughter cells that will differentiate into the appropriate cell type. Common adaptations include cell enlargement or hypertrophy by the addition of new cell components. For organs containing self-renewing cells, excessive hormone stimulation or workload increases organ mass and volume by hyperplasia, the production of greater number of cells. Examples would be the uterus during pregnancy and the thyroid gland in Graves' disease. Denervation of muscles, disuse or withdrawal of hormone stimulation results in cell atrophy or the loss of cell volume or number. Chronic mechanical or chemical stressors can cause cells to change their tissue-specific characteristics and develop metaplasia or an epithelial mesenchymal transition. This is unstable cell forms with abnormal function and the potential to be transformed to cancer. Dysplasia is another form of cell transition in which proper quality control measures of cell division are lost, thus producing cells that are abnormal in appearance and are also precancerous. Some of the most devastating health events result from a blood clot that occludes a blood vessel, either in the heart as an MI or in the brain as a stroke. Cells in the stroke core have the worst hypoxic ischemic insult with the following characteristics that can lead to cell death by necrosis. These are the rapid loss of ATP production, resulting in diminished membrane potential, the depressed sodium-potassium pump activity, the intracellular calcium overload, cell swelling and membrane rupture, the loss of antioxidant function, the accumulation of oxygen-derived free radicals, acidosis, the activation of degradative enzymes that can contribute to membrane rupture and spread the damage to adjacent cells. Apoptosis is a very common mechanism of cell death that contributes to normal cell turnover. Apoptosis or programmed cell death is due to the steps following an orderly sequence or death program in tissue disruption, organ dysfunction, and inflammation are minimized. Apoptosis can be initiated by extracellular signals like in extrinsic pathways that bind to the cell membrane tumor necrosis factors, causing an assembly of intracellular proteins that can activate the apoptic enzymes. Apoptosis can be initiated by intracellular signals such as the intrinsic pathways of abnormal cell functions that cause release of the enzyme cytochrome C from mitochondria. Cytochrome C then triggers a cascade of increases in pro-apoptic proteins that activate the apoptic enzymes. Normal, healthy aging is associated with decreased numbers and functions of cells. The program theory states that aging is a genetic program that has developed to direct senescence and death in order to benefit future generations of a species. The evolutionary theory argues that the forces of natural selection decline with aging due to accumulation of mutations. The disposable soma theory postulates that organisms have limited resources that have to be divided for maintenance and reproduction. The hyperfunction theory suggests that pathologic processes that lead to senescence are a result of gene overactivity rather than damage, breakdown, or failure. Senescence is a phenomenon that only occurs in cells that have the ability to replicate. Accumulation of senescent cells with aging is potentially a reflection of an increase in the conversion of cells to a senescent state or a decrease in the rate of death and clearance of cells. Telomere attrition is the mechanism of replicative senescence involving progressive shortening of telomeres, resulting in cell cycle arrest. With each round of replication, telomeres grow shorter, causing the chromosome ends to be less protected. Eventually, these are flagged as damaged DNA and signal cell cycle arrest. Genomic instability is when DNA alterations can potentially affect important genes and transcriptional pathways, thereby resulting in dysfunctional cells that, if they are not eliminated, may prevent an organism from carrying out necessary functions of survival. In aging organisms, this is important in stem cells because damaged progenitor cells can no longer participate in tissue renewal. Aging is linked to impaired protein homeostasis or proteostasis. This has two mechanisms maintaining proteins in correctly folded configurations and degrading proteins that have been misfolded. Deregulated nutrient sensing is accomplished by two major pathways that regulate metabolism. These are insulin and insulin-like growth factor 1 that signal the pathway and the sirtuins. Insulin is secreted by the pancreas, and IGF-1 is produced by the liver in response to the growth hormone that is secreted by the pituitary gland. In mitochondrial dysfunction, as cells age, the efficiency of the respiratory chain tends to decrease, which increases electron leakage and reduces the production of ATP. With stem cell exhaustion, as the tissues age, their ability to regenerate is significantly reduced due to the inability of stem cells to reproduce. With epigenetic changes, a variety of factors affect cells throughout the lifespan. These can include the DNA methylation patterns, post-transformal modification of histones, and chromosome remodeling. With altered cellular communication, neurohormonal signaling becomes deregulated in the setting of increased inflammatory reactions, decreased immunosurveillance against infectious agents and pre-malignant cells, and peri- and extracellular environmental changes.